Hi, welcome to video one of section A5, the nervous system. So we're looking at what a motor unit is, we're looking at what muscle spindles and Golgi tendon organs are and how they work, and we're looking at receptors. Chemo and baroreceptors we've already looked at, so it'll be a bit of revision, and thermoreceptors are new to us, and we'll continue to do work on those in later units as well. So let's start off with what a motor unit is. A motor unit, first of all, allows movement to occur in the body, and it also allows adjustments in movements. So it's made up from a motor neuron, which is shown here with this line. It's a nerve, that's all it is. It's a nerve that links the central nervous system, so our spinal cord and our brain, those nerves there. It links it to our skeletal muscle. You can see here it breaks out into little branches there, and each one has a motor end plate which allows it to be stuck to an individual muscle fibre. Do you remember looking at muscle fibres before when we looked at the size of them and how many are bundled together? And you can get smaller bundles or you can get large bundles. So a large motor unit is the motor neuron and all the muscle fibres that it attaches to. A large motor unit will have hundreds of attachments here. A small motor unit will have, well, maybe 4 like you can see here. Now the important things to remember here is that the muscle fibres are type 1, type 2A or type 2X. Go back to section A2 if you can't remember those types. So type 1 was a slow twitch fibre. You see where these little dots are here, where the motor neuron is attached to the muscle fibre to allow that electrical impulse to go through and contract the fibres. Well, one motor neuron cannot attach to multiple types of muscle fibre. So one motor neuron will be attached to only one type of fibre. But within this bundle of muscle fibres, you may have different types. So you will have another motor neuron that's coming down from a different part of the central nervous system that may attach to different different muscle fibres next to it if we have more muscle fibres here and actually it was a great big unit. And then we could say that these muscle fibres here are fast twitch fibres, let's say type 2A, the fast oxidative type, and these original ones here are attached to the type 1, the slow twitch. So each neuron is only attached to one type of muscle fibre. Okay, I hope that makes sense. So looking back onto here, when the motor neuron is stimulated, so when a message is sent down, an electrical message, then the muscle fibre that it is attached to contracts. When it contracts and shortens, we call that a concentric contraction. So when we're recruiting muscles for exercise, we're talking about which ones we're choosing for the exercise. So which ones are we choosing that are suitable for the exercise we are doing? So imagine this. If there was an empty box on the floor and it had a picture of a TV on it, and I said to you, oh, there's a TV over there, we've got a brand new TV for our room, please would you just go and pick it up and pop it up on the table so it's not going to get damaged, someone's left it on the floor. You go over, you go to pick up the TV. What would happen if there was no TV in there, if it was an empty box? Think about what would happen in that situation. You think something's heavy, you go to pick it up and it's light. So yeah, you're right. What would happen is you'd lift it far too vigorously. You'd be anticipating the requirement of a large force. You feel, your body thinks, your brain tells your muscles, oh, this is going to be a bit heavy, so you're going to recruit these muscles. If I told you the other way around, said, oh, don't worry, the box is empty, there's no TV in it, and you went over to lift the box, your your brain would be anticipating and sending electrical signals down to your muscle fibres to say, oh, it's all right, it's light. So you only need a few muscle fibres. So we'll recruit those small motor units. And you wouldn't be able to lift the box initially. But your body is so amazing that it would send those messages back going, oh, it's heavier than we thought. Can we recruit more units or larger units? And you would then adjust your muscle contraction to be able to lift the box. So it's really, really clever. So here, look, we've got the body is able to make those adjustments, and this is how it does it. First of all, it chooses different fibre types to recruit. Do we need the slow twitch? Because is this going to be a low level force that maybe we'll need to repeat our actions a lot? Or will we need the fast twitch, which is really powerful and we're only going to have to lift something once? If you remember the recruitment ladder, in actual fact we don't just ever recruit those fast twitch, we recruit all types in that situation, but maybe a high percentage of those fast twitch fibres. We may look at the frequency of stimulation, so how many electrical impulses are we going to send from the central nervous system down to those muscle fibres? Are we going to keep that stimulation going? As in, do we need to keep those muscles contracted for a period of time? And then we've got the different size units. So do we recruit the motor neuron that was attached to 500 fibres within our quadricep, let's say? Or is it a smaller amount where we just need to use the motor neuron that was attached to 3 or 4 fibres? So we can look at different fibre types, frequency of stimulation of those muscle fibres, and different sizes of motor units. So you've got a task now. Please could you stop this video and can you write down, referring to the 3 bullet points above, how you would apply those to 2 varying sporting situations? So not lifting a television, but potentially changing the response in a tennis match. So you're going to do a net shot rather than a smash shot. You are going to make a short pass in football as opposed to a lofted pass over the top. How do you change or vary your output via or using muscle fibre recruitment? So you should have done that now for me, and this would be a mark scheme, a typical mark scheme that you would see. So activities requiring high levels of force, and you must have used a sporting example that requires high level of force to actually get some of these marks, because that's what the question asked. So you could have said type 2A or type 2X would be recruited. If it was very high force, we'd expect to see 2X. If it was moderate to high force for a longer period of time, like marking somebody for a minute or so, we'd expect to see 2A. We're looking at high frequency of firing or stimulation and large motor units. And then the opposite, again, you needed to have an example from a sport that uses or requires less force, and we'd be looking at slow twitch fibres or type 1, less frequent firing of the neurons, and smaller motor units would have been recruited. Muscle spindles are what we call proprioceptors. So they are found within the muscle. Now my picture is not wonderful here, but you can see it's like a little slinky I've drawn there, and it's wound around those muscle fibres. That's called a muscle spindle. As the muscle stretches out, as you lengthen the muscle when you stretch, this spindle, or it looks like a slinky, will stretch out as well. And when you contract your muscle, the spindle will contract. Now this spindle can therefore detect the length of those muscles. So now for me, if you close your eyes and you extend and you flex your arm, your body will be telling your brain the movement of those muscles. If you were to close your eyes and somebody else was to move for you, so you were not initiating the movement but someone was moving your arm around, you'd still be able to tell whether your arm is, whether it was extended, whether it was flexed, whether it was rotating. And this information comes from those spindles. It tells you about the change in length of the muscles. And actually, it can tell you about the speed in which that muscle lengthens or shortens as well, which can be very important in sport. So why do we have them? Well, we have them for adjustment because we need to know how far we need to stretch. The body tells us how far we're stretching, and so we can reduce it if we need to. So it's a way of talking. The initial impulse comes down the motor neuron here to tell the fibres to contract, and this spindle sends messages back up via the sensory nerve to the central nervous system to say, oh, it's moved by this much and this is how fast it's moving. But it has a safety aspect to it as well, which is that it prevents the overstretch injuries. So this is what happens. You stretch your muscle too far and too fast, and a stretch reflex is initiated. So the spindle sends this emergency message up going, hang on a minute, you are going to hurt yourself. You're stretching too far and this is going too fast for me to handle. We're going to end up tearing the muscles. So it's an involuntary, as in we don't have any say in it. This happens automatically inside our body to protect us. So the involuntary response of, or initiated by the muscle spindle, so a response of the nervous system, is to cause a concentric contraction of that skeletal muscle. So it suddenly shortens. Apologies for my overreaction with the slide there, got a bit excited. Let's move on. So how does it work? The spindles detect the change in length and the change in rate, so the speed of that extension of the muscle, of that stretch in the muscle. It reports via the sensory nerve everything to the central nervous system, and the central nervous system was our spinal cord and our brain. This mechanism, this muscle spindle, is used to adjust movements. So if you are walking along a beam, for example, it will constantly report back to the central nervous system about where the muscles are and how they're contracting, how fast and how far they're moving, and will allow you to send different muscles out to say, oh, a bit more or a bit less, so you can stay balanced, for example. It also prevents you from damaging yourself by overstretching, so it sends that emergency involuntary message to the CNS if you've got a very big stretch that's happening very quickly. Now you can do a little experiment on this where you cross your legs and the top leg needs to be fully relaxed, and use the side of your hand to hit just below your patella. You can, if you push it with your fingers first, you can feel it's quite fleshy, and if you hit that, it stretches the muscle quite quickly and will give you a stretch reflex. And the stretch reflex is a contraction of the quadricep muscle, so a contraction of the stretch muscle. And that contraction obviously prevents it from being overstretched. So it's really clever. So it receives the message about the state of the stretch. The CNS sends a message again via the motor neuron, but it's a different message to last time, to contract the muscle this time and prevent it from overstretching. It's an involuntary contraction, which means that we don't have any say in it. It happens without our knowledge. It's our body's safety mechanism, and it's called a stretch reflex. But we also use this system, but not the emergency version of the system, if you like, to vary the muscle contractions for our sporting actions. So another proprioceptor in the body is the Golgi tendon organs, and you can see these here, and they are in the tendons of the skeletal muscle, and they're slightly different. They're not slinky shaped, and they're slightly different because instead of checking the rate of length in the muscle and the length of the muscle, it's now looking at tension within that muscle. So it's now not looking at how far we've stretched, but how much force we're trying to apply, essentially. So they're found in the tendons and they detect tension in the muscle. But the messaging system works exactly the same way as the spindles. It sends messages from the muscles up to the CNS via this sensory nerve. And then if we need to adjust, we will send a message back down the motor neuron to contract and recruit different muscle fibres to suit the activity. So the information is used to adjust the amount of force applied by maybe taking smaller motor units, bigger motor units, increasing the firing towards those motor units, or picking larger or smaller motor neurons. If too much tension is on the muscle, so I've asked you to pick up that TV and it's way too heavy for you and you're really, really struggling, our body again will give you an involuntary response to it. But this time, instead of contraction of the muscle, it will relax the muscle for you. So you've lifted that TV, it's really, really too heavy, and you drop it. If you think of those weightlifters, the Olympic weightlifters that are lifting huge amounts of weights, they lift it up above their head and they can drop it, their muscles just give way. And that can be an involuntary relaxation of the muscles. Sometimes they actually pass out if you watch them, and that will come up in our next topic area. So this again, these Golgi tendon organs, they adjust the muscle fibre contractions to suit the type of contraction we need for the activity. So they can be used for adjustments and movements, but they also protect from muscle tears as well by relaxing the muscle when it thinks you're going to tear a muscle because you're holding something that's too heavy. So our last section here is to look at receptors in section A5, and these receptors are found in the aorta and the carotid artery, these chemoreceptors and baroreceptors, which we've talked about before. Now these receptors are obviously linked with nerves up to the brain, and I've just got this little diagram for you here, so it shows you where the medulla oblongata is up here, and we're going to come to the hypothalamus in a little minute, which is at the top. And I thought you might be interested to know that this section here is called the brainstem, and it's spoken about quite a lot. It's a very important part of the brain. So this is revision essentially. Chemoreceptors are located in the aorta and the carotid artery. So the carotid artery is the big artery that you can take your pulse with on the side of your neck, and the aorta is that lovely big artery coming out of your heart from your left ventricle. They detect a change in pH, and if we're exercising, therefore they detect acidity, which would be caused by an increase in CO2 and hydrogen ions. It sends nervous impulses to the respiratory control centre, and as a response increases breathing rate and depth. It can also impact heart rate and increase heart rate and stroke volume. The baroreceptors are located again in the aorta and the carotid artery. These guys detect pressure changes, and the pressure changes, just to refresh our memories, could be because of increased cardiac output and could possibly be dehydration. Cardiac output meant more blood was coming out of the heart over 1 minute, so stroke volume has increased. That amount of blood coming out of the left ventricle in one pump, and possible dehydration would cause resistance. So blood pressure was cardiac output times resistance, and that dehydration, that making the blood really thick, can cause resistance and therefore up the blood pressure. Both occur when we're exercising. So it stimulates the sympathetic nervous system. The sympathetic nervous system gets things done. It's the active nervous system. It's the thing that speeds things up and makes things work. So if the pressure changes, the sympathetic nervous system can dilate vessels if the pressure goes up, and they can constrict vessels if the pressure goes down, to keep your blood pressure constant, because this is what it wants and what it needs. So just take a couple of minutes to have a read through this section for me, please, if you can pause the video. And if there's anything in here that you cannot remember, you're not sure about, please can you go back to previous sections on the cardiovascular system, A4, and check that video out again, please, because this should all be making sense to you now. So finally, our last receptor is a thermoreceptor. Now I'm sure you can figure out what that is straight away from the word thermo. And it is, and you are right, a change in the core body temperature. So not skin temperature, not environment temperature, but the core body temperature, what's happening actually inside the body. These receptors are found in the skin and the hypothalamus. Now they're found, and this is more than we need to know, but in the skin they're found sometimes near the surface and sometimes deep within, depending on what they're trying to detect, heat or cold, if you like, hot or cold conditions. But together, with each other, they do detect temperature of the blood, and they relay messages via the hypothalamus, and the hypothalamus has receptors in there as well as being the organiser and the management area of it as well. So when you exercise, muscle contraction and energy production in those muscles and friction of the muscle fibres rubbing together when they're working causes heat to be generated. That heat has to get out somehow, so it gets transferred into the blood, and the blood temperature increases. And we have to regulate that. We cannot have a hot blood system going through. We need to cool it down. So our body responds when this thermoreceptor picks up that we've got a significant increase in core body temperature. It causes our vessels at the skin to dilate, which means that the heat can be released through the skin. If we're in different conditions, if we're in cold conditions, so, and this isn't a sporting example, but if we fell into the sea, for example, and it's cold, obviously it can kill you. So what our body would respond by doing is constricting our vessels, vasoconstriction, to prevent the heat from being lost so we can stay alive. So vasodilation is the response to exercise, and it's vasodilation of the vessels at the skin, and the aim of that is to cool. We do a lot more of that in section F towards the end. That's it for now.